Investors, the potential of egg yolk and whey milk is 100% as far as glutathione is concerned, glutathione potential. Cysteine contains sulfur and is formed from two molecules of cysteine. Cysteine is also a sulfur-containing amino acid used to manufacture glutathione and taurine. You have heard about glutathione, right? You have heard also about taurine. Okay. Cysteine is a non-essential amino acid that is synthesized by the body. Cysteine possesses life extension potential by participating in the repair of some forms of DNA within the body. Cysteine plays an important role in the communication between immune system cells, that is signaling, thus enhancing immune system function and maintaining optimal health. Cysteine also promotes the activity of white blood cells. Cysteine stabilizes extracellular proteins. Cysteine is a powerful antioxidant that deactivates very powerful reactive oxygen species like hydrogen peroxide, free radicals, hydroxyl free radicals, singlet oxygen free radicals, and superoxide free radicals. Cysteine interacts with aldehydes and prevents them from initiating cross-linking. Cross-linking leads to rapid aging and wrinkled skin. Cysteine deficiency can lead to reduced production of the essential peptide glutathione, which acts as an intracellular detoxifying agent and is important in maintaining hepatic function. The role of glutathione is like chelator. It chelates toxic metals and throws it away. That's like EDTA. Here, the glutathione, you know that? Glutathione is known to increase the integrity of telomeres. Okay, you have there the chromosome, you have there the tail, the telomeres. Telomeres shorten due to damage by free radicals. If the telomeres shorten, the lifespan, your lifespan is shortened. The lifespan of the cell is shortened. Free radicals play a role in DNA mutation, and there is evidence that mutations in your telomeres can cause larger chunks than normal to be lost during cell division. That means glutathione can extend life. Lack of glutathione shortens lifespan. And what is the best source of glutathione? Egg yolk. Because it's very resistant and very milk. Glutathione plays a role in nutrient metabolism and regulation of cellular events. For gene expression, DNA and protein synthesis, so stem cell enhancing, cell growth, stem cell replication, immune response, system detoxification. Eating glutathione has negligible effects on our health, for it will just be broken down quickly in our digestive tract and eliminated. The best way to raise your glutathione is to provide the body's building block, precursor it needs, to manufacture glutathione for itself. So the best way, you have to eat a lot of egg yolk, you have to drink a lot of whey milk. Carnosine, I discussed that earlier, is a combination of two important amino acids, beta-alanine and L-histidine. It is called the miracle worker due to its ability to change into numerous compounds that can each carry out or enhance many important body functions. All in one super antioxidant, effectively quenching the most destructive of free radicals: hydroxy radicals, superoxide, singlet oxygen, and peroxy radicals. It has the remarkable ability to actually rejuvenate cells. Okay? Approaching senescence, the end of life cycle of dividing cells, restoring normal appearance and extending their lifespan. Where do you get carnosine? You get that from meat. Helps prevent skin collagen cross-linking, which leads to wrinkles, which leads to loss of elasticity, wrinkles, macromolecular disorganization, and loss of extracellular matrix. Prevents glycation. Glycation is when you are diabetic, you have very high blood sugar. The sugar binds with protein to form glycated protein, and that is highly oxidizing. It is so destructive. That's why it can destroy anything in a diabetic patient. Powerful than vitamin D. Astaxanthin is 550 times more powerful than vitamin E and 11 times more powerful than beta-carotene at neutralizing singlet oxygen. It is a powerful ultraviolet B absorber and reduces DNA damage. Astaxanthin molecules protect the water-soluble part of the cell as well as the fat soluble part of the cell, so it can protect the entire cell. It is a very natural anti-inflammatory. Astaxanthin crosses the blood-brain barrier and the blood-retinal barrier, beta-carotene and lycopene do not, which brings antioxidant and anti-inflammatory protection to your eyes, the brain, the central nervous system, and reducing your risk of cataract, macular degeneration, blindness, dementia, and Alzheimer's disease. Astaxanthin improves athletic performance and induces better brain health. Fish, due to their red pigmentation, are great sources of astaxanthins, and these are the rainbow trout, sockeye salmon, pink salmon, and red bean. Fruits and vegetables with red coloration, red peppers, carrots, tomatoes, radishes, and other fruits and vegetables are also very high in astaxanthin. Now let's move now to what I am doing. Cellular and biochemical medicine, the natural and comprehensive approach to enhance stem cells. Principles behind this practice of mine. Living organisms require the constant expenditure of energy to maintain their complex structures and processes. Central to life processes are chemical reactions that are coupled so that the energy released by one reaction serves as a substrate for the second and so on. So it's like a chain reaction. Chemical reactions are directed along specific sequences called metabolic pathways. These reactions are catalyzed by enzymes. The enzymes are protein catalysts. It speeds up reaction. And each participant compound is called a metabolite. The many thousands of different types of biochemical reactions within a cell do not occur independently of others. They are rather linked together by intricate webs of interrelationship, the total pattern of which constitutes cellular organism. Chemical reactions can be speeded up by increasing the average kinetic energy of the reactants through catalysis performed by a class of protein molecules called enzyme. Without the assistance of enzymes, most of the chemical reactions of metabolism would barely proceed at all. So it's very important that you know what enzyme to use so that you can speed up stem cell replication, stem cell maturation, stem cell reduction. Without, I said earlier, with enzymes you can do that within hours, within minutes, within days, or within weeks or months. But without enzyme, it will take you years and you will not get the desired results. All nutrients that I discuss must be present in the diet in order for the body to maintain basic life processes. Okay? But each nutrient has its own specific functions and relationship to the body. But remember, no nutrient acts independently of other nutrients. It won't work if you say, I just give vitamin C, so okay, it won't work. It won't work if you say, I just give vitamin A or vitamin. No, everything must be there so that so that everything you can enhance the stem cell. And all nutrients, before they can serve their functions, undergo various changes or biochemical reaction. Basically, our body needs most of the following. Okay? For stem cell enhancing, enzymes, because they are biological catalysts, functioning to increase the rate of chemical reaction. Most enzymes are proteins and their catalytic action depends on their protein structure. Coenzymes and cofactors are ions and molecules that were removed in the purification procedure but needed for enzymatic activity. Coenzymes are organic molecules that are derived from water-soluble vitamins. Coenzymes participate in enzyme catalyzed reaction by transporting hydrogen atoms or electrons and small molecules from one enzyme to another. And these cofactors are metal ions such as calcium ion, magnesium ion, manganese ion, copper ion, or zinc ion. Cellular biochemical medicine offers a complete approach to enhance stem cells through rapid cellular balancing. That's what I use. As stem cell enhancer, it has the capacity to support the natural release of adult stem cells from the bone marrow, which migrate throughout the body and support the natural renewal processes of organs and tissues. So it can differentiate. Number 2, diet modification. The diet should be 40% protein, 40% fat, and 20% carbohydrates, mostly coming from fruits and vegetables, carbohydrates. Exercise and rest. These are often forgotten. We either have enough rest or we lack rest. RCB uses biochemical salts. Major and minor minerals, enzyme cofactors, coenzymes, essential amino acids like methionine, lysine, chain breaking and membrane stabilizing antioxidant. So antioxidant that will stabilize the cell membrane, antioxidant that will break what you call a chain reaction of ROS, which will damage the whole body, to enhance the body's energy producing metabolism in the cells, known as the Krebs cycle and electron transport chain. The Krebs cycle was developed by, by the Hans Schroeder from Germany. Is a Nobel Prize, and the electron transport chain was developed by nothing else by Warburg. Okay. Stages of energy producing metabolism in the cell. There are 3 stages of energy producing metabolism in the cell, and these are the following. The first stage is anaerobic stage called glycolysis. So there are 2 stages. One is anaerobic, in the absence of oxygen, and one is aerobic, but this is divided into 3. Sugar, glucose, is broken down to energy in the absence of oxygen, and that is what we call anaerobic glycolysis. Okay. And then the next 2 are aerobic stages of metabolism. Second stage is called the Krebs cycle. Okay. And the third stage is the respiratory chain. Warburg's. The first step of energy producing metabolism, which is anaerobic, it produces 2 moles of ATP. ATP is the currency of energy in the cell per molecule of glucose. So 2 moles of ATP per molecule of glucose. That is in the absence of oxygen. The primary product of glycolysis when the aerobic states are not operating is lactic acid. And sometimes alcohol, but not pyruvate. So what will happen to the surrounding of the cell? The surrounding of the cell will become acid, right? Now, if there are normal cells having too much acid, the cells will dedifferentiate. Your normal cells will go back to stem cell status and become cancer cells. So they dedifferentiate, so stabilize. And anaerobic metabolism is the single cause or essential requirement that turns normal cells dependent on aerobic metabolism into cancer cells. So this is what happens. If the normal cells, because they are designed to breathe in the presence of oxygen, can no longer breathe because they are surrounded by lactic acid, so what will the cells do? It has to find a way to escape. It has to survive. So the normal cells will go back to stem cell status. That's why when you examine cancer cells, you look for embryonic antigen and fetal antigen. For liver damage, you look for alpha fetoprotein. So that is the protein of the fetus. But you're dealing with a normal adult. And for colon cancer, you look for carcinoembryonic antigen. So all the antigens that you get are stem cell antigen. You understand, right? Because the cells could not survive the acidic medium, it is the DNA can dedifferentiate and produce cells that will not die in acidic medium. You understand, right? So, how would you control this? So I look at this. This is glycolysis. Okay. The first step is we have the glucose. Okay, 6 carbon molecule is converted to glyceraldehyde 3-phosphate with the loss of 2 moles of ATP loss. Okay. Then it's further converted into the glyceraldehyde phosphates to the work of the niacin as nicotinamide adenine dinucleotide is converted to